Good morning and welcome everybody. I'm Jay Leffler. I am the Chief of Radiation Oncology at the Mass General, and we're so excited to be hosting this webinar. Joined by three distinguished colleagues of mine, who I will introduce briefly. Today, the focus is on clinical innovation, clinical research. It's been a very trying year, as you'll hear. And we'll hear about the impact of the pandemic on our ability to do cancer clinical research. Just some housekeeping issues. No microphone or video capabilities are needed. If you have questions, we would like you to submit them to the Q&A button in the bottom of your screen. The webinar, as you've just probably heard, is being recorded, so it'll be available to others who either haven't been here but wanted to come or were late to join us. As I said, today we'll be focusing on clinical innovation, and I'm very excited to be joined by three distinguished clinical and research innovators. Dr. Keith Flaherty. Dr. Flaherty is the Richard Saltonstall Chair of Oncology at the Mass General. He's Director of Clinical Research at the Mass General Cancer Center. He's a professor of medicine at Harvard Medical School. But I think what you'll really be interested in, he's also put on another hat this year. He directed clinical research for the COVID pandemic, and his work was invaluable for the institution to deal with this most horrible situation. So welcome, Keith. Becca Heist is you'll pick her out because she's the only woman on the panel. Welcome, Becca. Becca is the Associate Director of Clinical Research at the Mass General Cancer Center, and she's an Associate Professor of Medicine at Harvard Medical School. And last but not least is Richard Hoden. Richard and I work together a lot. He's a very distinguished endocrine surgeon, Director of Endocrine and Surgical Oncology. He's academic dean for the Department of Surgery. He's a professor of surgery at Harvard Medical School. And I can tell you, if you have patients with endocrine problems that need surgery, he is one of the top people in this country, if not the world. So I'd like to turn things over to Dr. Flaherty, who's is that your living room, Keith?
This is a library.
A library, okay.
Yeah, it's got too many books to count as a living room.
Okay, so it's my pleasure to turn things over to Dr. Keith Flaherty. Keith?
Thanks, Jay. So thank you all for joining us this morning. I'm really excited to hear Becca and Rich's comments, but I thought I would maybe tee things up by briefly touching on two areas in cancer, which is what we're mostly here to think about, deliberate on this morning. But also to Jay's point, maybe I'll make a comment about COVID research at Mass General, which I have had the pleasure of being involved with, the honor of being tapped to direct the clinical research effort in COVID, and I think it's actually related to some of the points that we're gonna cover today. So I'll maybe put that in the third position. It's kind of a transition point. The comment that I always make, well, certainly when I'm talking to other doctors who are particularly those who are in their training years and are considering where to think about joining faculty, they're interested in doing research, particularly around new therapies in cancer. When I talk to them about what makes Mass General special, I mean, kind of use some of those talking points a little bit this morning, because I think they're highly relevant to why patients come to Mass General for their cancer care, but it's behind the scenes, and I think these are kind of recruiting points that I use, more for physicians in some ways. And the first one is that we are embedded in the country's largest research environment, and that's that there's two or maybe even three levels to that. One is Mass General, as you may know. Certainly, if you live in the Boston metro area, or perhaps even beyond, you've seen billboards or signage even that Mass General advertising that point that we are the country's largest research hospital. That's by funding dollars. So yes, we have many employees, but it's the funding that they command and receive that is what's behind that statement. So we are, by any measure and that measure, just a massive research environment, and it's highly relevant for cancer, as I'll come to in just a moment. And then beyond that, of course, we're part of the Harvard University system of hospitals and research institutions. That's you get another layer. Just across the river is MIT, and then as many of you who are active in the private sector know, Boston and Cambridge are the largest biomedical research community in the world. There's never been anything like it, in fact. I came to Mass General 11 years ago when it was already pretty established, but it's rather amazing the degree to which that community and that ecosystem has grown further. And why do I start with that? Because it really is important to the point that that if you're going to try to advance treatments for cancer patients, it really it must be embedded in cancer science, and has to be embedded in the very best cancer science, the most robust, not just a clever, unique idea, but truly what we have reason to believe are the most important ideas. And we're in an environment that allows us, I think, to choose wisely in that way. We have actually led the development from early phase clinical trials, phase 1 clinical trials, through to FDA approval of the largest number of therapies of any cancer research program in the country. Certainly over these past 11 years that I've been at Mass General, that holds true. And it's, I would say it's really a consequence of this environment that we're in, the partnership that we clinical investigators have with our scientific investigators. So let me transition to a couple vignettes briefly, one of which Becca has been involved with in the more recent years. And I'm not going to steal her thunder by touching on that example if she'd like to comment on herself, but I think it's the story of a drug that was just recently FDA approved, for which we dosed the very first human beings in the phase 1 trial. And this is a drug that's in a class of drugs referred to as antibody drug conjugates. In other words, there's a monoclonal antibody, which is a naturally occurring immune system molecule, re-engineered to latch onto a certain protein on the surface of certain cells and certain cancer cells, it turns out. And this is an insight that was gained maybe even 10 years ago, roughly, that this surface protein is overexpressed commonly in breast cancer, a certain form of breast cancer, lung cancer, and certain genitourinary cancers, including bladder cancer, amongst others. And as a company that was thinking about developing this program, a pretty small company, came to Mass General 7 years ago to meet with us to talk about kind of the conception of how to think about developing this drug. We sat with them, met with them, and really mapped out all of the paths that we saw for it in terms of which cancer types we thought were really potentially the most addressable, where this therapy might have the most impact, and advising them to put most emphasis there. And then, of course, orchestrating our efforts internally to recruit those patients primarily. And breast cancer was certainly at the top of the list, at a minimum tied for the top of the list. So we led that trial and pretty quickly uncovered that it was going to be a quote unquote breakthrough therapy. That's actually a real designation that the FDA now uses for cancer therapies that show great promise in their first human study. And so that ultimately linked up to FDA approval in breast cancer, and our lead investigator in breast cancer in our early drug development program, Aditya Bardia, who led that phase 1 trial, also led the phase 3 trial. So it was a straight through line, if you will, in the example of breast cancer. But the way that we're organized is as a cancer center is to be able to pursue all of those other opportunities to really try to get a promising therapy to the largest number of patients who could benefit. And it is a bit of a unique culture and dynamic that we have, such that Becca Heist, who effectively sits right next to Aditya Bardia in our early drug development group, she was there from the beginning to lead the development of this agent in lung cancer, where it is making rapid advances as we speak, and we've been investigating in other cancers beyond that. And it's again part of the concept or the theme of following the science, but also really simply trying to serve our patients here and now, and then of course make advances for all the patients that we're going to be seeing over the years to come, to try to advance the field as quickly as possible for them. But the thing I wanted to land on in this example that to me is just so inspiring, and really is just, it speaks to the environment in which we operate and what makes Mass General as a cancer center unique, is that this drug, this so-called antibody drug conjugate, it delivers the treatment on the back end of the antibody. That's how it's designed. And this is an emerging, and I would say actually exploding class of new cancer therapies. And you can imagine that if there are cancer cells that could resist the effect of this drug, there might be two ways they could do it. One is that they would figure out a way to no longer express on their surface the molecule that the antibody component is directed to. So that's one problem that we anticipated actually even before the first patients were ever treated. But it turns out through parallel scientific research that Leif Ellison, who's a research partner of Aditya Bardia's, he's head of the breast cancer group overall at Mass General, but he has a laboratory in which he began investigating that potential problem. How might cancer cells lose the surface receptor, and was there a way to restore that? Are there second therapies that you could use to actually prop up the receptor expression and make this drug potentially more effective for patients, maybe even extend its use to other cancer patients entirely? But what he learned early on in that research is that the drug that's delivered on the back end of the molecule, that there's unique resistance to that component as well. And that's one that we did not anticipate going into this endeavor even 6 years ago when we first dosed the very first patient. And now what's happened is that Aditya and Leif have partnered and developed a research program that looked basically at both ends of the problem, which is to say that they are not only trying to anticipate what might be resistance mechanisms, but head that off with therapies that would basically overcome that resistance. And this is what we call ultimately rational combination therapy. So if we take a therapy into our program, not only are we trying to get it out to all the patients who might benefit from it and know at a molecular level who those patients might be, but really also how to then see that therapy through to have maximal impact by anticipating what might be the combination approaches. So that's an example. We've got a bunch of others that are just like that. But let me just move on to another vignette that I thought would be a useful point of introduction, at least. And that is the work that we've done in what we call the immunotherapy toxicity program at Mass General. You're probably all aware of the fact that there's been this revolution in terms of a new class of therapies that motivate the immune system to attack cancer. And the most successful of those have now gained FDA approval and across many cancer types and counting. But there's a problem, which is that these same drugs can activate the immune system to attack normal parts of the body. And that's a pretty common problem, as it turns out, and can be even a devastating problem. And the field has really lacked insight in terms of understanding the difference between the immune system being activated to attack the tumor versus normal tissue, and how you might predict that. And at a minimum, how you would monitor that in a way to be able to disrupt the dangerous component of that, the attack of normal tissue, and ideally preserve the anti-tumor component. So we built a program to clinically manage this, the consequences of these therapies for those patients who had the most severe complications. And that led to the establishment of the country's first severe immunotherapy toxicity service in the hospital, which Kerry Reynolds one of our close colleagues, established. But immediately, as that program was forming, about 40 faculty from across Mass General, who cut across all sorts of disciplines outside of oncology, who were experts in inflammation, immunology, autoimmune diseases that could affect various parts of the body. Those experts already existed in Mass General. That's the beauty of doing cancer research in this environment, and taking care of cancer patients in this environment, is that when we recognize the effect of these therapies, and we pose the question, how can we better manage these patients, more safely manage them? And how can we actually conceive of the idea of doing research so that we can improve the anti-cancer effect while trying to pare back or eliminate the dangerous side effect side. And that program now has been fully operational for 3 or 4 years now, and reflects just another example where being embedded in this environment has such power. So I'll just close by saying that that the COVID work has been entirely analogous to that. What has made COVID research at MGH powerful is our ability, unique ability, to tap into this very large community of researchers who have varied expertise, of course, in virus immunology, but inflammation, and then some of the organs that are damaged or involved by COVID. Also, we're able to quickly, and I mean within a week's time, rally around a research program, a coherent research program, the likes of which had never existed, not only at Mass General, but in the country and in the world, because there had been no other virus, including influenza, that quite caused the same spectrum of problems. So it's been really a pleasure to take this concept that we've really hammered out well in oncology and bring it to bear in COVID, and that really has been simply the secret behind that work. So we can touch on that perhaps a little bit later, that overall concept. But I'd like to keep moving. And so in the order on my screen, we'll move over to Becca Heist, who Jay's already introduced, for her comments.
Thanks, Keith. So, as I think about all of the advances that clinical research and scientific basic science research has enabled for cancer treatment, I think really, there's almost no better place to tell that story than in the treatment of lung cancer. And, when I think back to about 10, 15 years ago, when we were sitting with a patient newly diagnosed with lung cancer, the conversation was really about chemotherapy and how to choose chemotherapy based on whether it was non-small cell or small cell. And in reality, we were choosing based on what we thought the patient could best tolerate, what kind of toxicity we were avoiding. And the outcomes were frankly just not very good. And since that time, when I sit in front of a patient newly diagnosed with lung cancer now, the conversation is so much more complex. There is so much better treatment, and the prognosis is so much better. And it's really a sea change in terms of how people are treated and how they do. And that all has been driven by research. And, as an MGHer, I'm proud to say that much of that research has really come through and out of MGH through the collaboration and that community of researchers that Keith was talking about. So just to take a step back in terms of the advances that have been made, now when we think about lung cancer, it's not just non-small cell and small cell. We really want to know, what's the histology within non-small cell? And then even more than that, what's the molecular profile? What's the tumor genomics? And what's the immunotherapy profile? And to just get into that a little bit further, I'm going to talk about two broad things, targeted therapy and immunotherapy, and how research in both of those areas have really made an impact for people. So in targeted therapy, we really think about, I think the sentinel discovery was really out of Dr. Daniel Haber's lab when with EGFR, which is a gene that can drive some lung cancers, he really asked, why do some people respond to these EGFR inhibitors, and what in their cancer is making them respond? And the finding that there was a genetic mutation within this gene of EGFR within the cancer in some patients, where if you had it, you would respond with kind of exquisite responsiveness to some of these EGFR inhibitors, really kind of opened up the whole targeted therapy world. And since that time, it's been this search for other mutations like that, other targets where if you have a mutation in the gene in that cancer, there's a drug that can really target that pathway and cause the cancer growth that activation of that gene is causing to stop. And in lung cancer now, we have at least 7 different targets. So when we're thinking about lung cancer, we're asking, is there EGFR, is there ALK, is there ROS, is there MET? And we want to know this because if there is, then we really don't want to start with chemotherapy. We want to start with a targeted therapy that actually targets that gene, stops that gene from signaling, and can kill cancer cells because of that. And I think one of the things as we think about all of the FDA approvals of targeted therapies that have come through in lung cancer over the past 5 to 10 years that are really changing how we treat people with a pill to start off with if they have one of these mutations. You know, we have to appreciate the research that went into that. And I can still remember in the early days when Jeff Engelman, who used to be at our lung cancer center, was talking about doing rebiopsies of people. And this was a concept that was kind of really just shrugged off for decades because it was thought to be high risk. But with the collaboration of our interventional radiologists, with their pathologists, with our patients, and with the science labs, we could really start to study, you know, when people were responding to these drugs, if disease was growing again, what was happening, why was that happening, and we really needed tissue to know and understand that. And that collaborative effort has really kind of moved the field forward in terms of understanding. Great, so the target works, the drug works, and then when it stops working, what do we do next? And all of that research within our clinical trials, within our translational research and labs has moved the treatment forward so that people with targeted therapies now, you start with one therapy. When you understand why their cancer is becoming resistant, then you move to a next therapy where the drug can possibly target that resistance or a combination therapy. And the whole field has been upended so that chemo is really kind of much later in the lines of therapy. And that's been a huge sea change for many of our patients. Similarly, I think there's another revolution that's happened in lung cancer where immunotherapy, which Keith touched on before, has really dramatically changed what we think about what's possible for our patients in terms of long-term prognosis and our understanding of how actually the cancer works and evades the immune system. And so now, you know, the conversation is targeted therapy, chemotherapy, and what about immunotherapy? And immunotherapy really has become the new backbone of our first-line treatments for lung cancer. And much of the work that's going on now with Justin Gaynor and all of the people in the labs who are focused on immunoprofiling these cancers to understand them better is really trying to figure out, great, so, you know, a certain proportion of people do wonderfully well with immunotherapy. They have great responses that can last a very long time. But for the people who are not getting those responses, how do we convert their tumors so that the tumor does become activated and become primed to get recognized by the immune system and benefit from what immunotherapy has been able to do? So now, you know, when I sit in front of a patient and talk about the therapies that are available to them, there's just so much more on the table. And what the therapies do are so much more beneficial for people than what they historically have been. And it is an amazing advance in how people with lung cancer are able to be treated that really has been driven by research. None of this would have been possible without the clinical trials and the basic research that gets done to drive these treatments forward. And it's an amazing feeling to sit with the patient who actually was on the study that got a drug approved, and they're still doing great on that study. And that kind of innovation and movement forward of the field is something that we all are extremely committed to. In addition to my kind of living in lung cancer land, I also work closely with Dan Urich and Keith in the Tremere Center, which really has brought first-in-human studies to patients. And I think one of the amazing things about the Tremere Center, and the Tremere Center is an example of this collaborative spirit. You heard from Keith about the collaboration that Aditya and I had. There are investigators from every single disease group that make up the Tremere Center, and we work together. And for just two quick examples of that, you know, the latest target that's hitting lung cancer and that will be a huge game changer is KRAS. And KRAS takes up about a quarter of lung cancers. G12C is probably a good proportion of that quarter. And for the first time ever, this was thought to be a target that was not druggable. And for the first time ever, there are very effective G12C inhibitors. And I participate now in these working groups with lung cancer docs, GI docs, basic scientists, all focused on G12C, trying to figure out what can we learn from the clinical trials? What can we learn from the biopsies that get done, from the blood biopsies that get done, the liquid biopsies that get done, to understand who's responding and who's not responding, and how can we get the people who are not responding to respond with other combinations. And that collaboration is something that I think speaks to kind of the spirit of how research happens at Mass General. We all are interested in helping our patients do better. And so we pool all of the resources and the work that we do so that we can better understand what is happening on these new targeted therapies. And I think G12C will likely be the next FDA approved therapy that's coming along. And there's work to be done because as with all of these treatments, it's never quite good enough, and we want to do better. But going back to the Tremere Center, one thing that really struck me recently, I was in a meeting with Dan Urich, who is with Keith, the leaders of the Tremere Center. You know, historically, first in human studies had a kind of bad reputation. They were thought, even at some point decades ago, to be, you know, is this ethical to do? Is there any benefit to the patient? And what struck me in what Dan was presenting was, you know, if you look at the patients who are coming through the Tremere over the last 5 years, the clinical benefit rate was remarkable. 1 in 3 patients had what's called a PR, which is a partial response. That's a substantial shrinkage of tumor burden. That's really what every drug really wants to achieve, or a stable disease that lasts at least 6 months. And so when we think about are we helping our patients, which is ultimately what we want to be doing? Absolutely, with getting the right drugs, the better drugs, the drugs that make scientific sense and that are kind of selecting the patients where it's most likely to have benefit. First in human studies are absolutely benefiting patients. And the clinical research enterprise is really, I think, what's driving the field forward in terms of being able to offer better prognosis for our patients. So with that, I'm going to close and I'll hand it over to Rich for his comments.
Great. Thank you, Becca. And thanks to everyone for joining us today. I'm really excited to talk a little bit about surgery and some of the advances that are being made generally in the field and specifically that we're working on at MGH. You know, with surgery, most people think about what we do in the operating room, and obviously that's important, maybe the most important thing, but we think of it as coming in 3 phases, what we do before, pre-op, and then intra-op, and then also post-op, and that all 3 phases are really important. And I thought I'd just touch on a couple of areas of exciting advances and research in all 3 phases of surgery. So in terms of pre-op, you know, when we think of cancer surgery, I mean, for many, many decades, really since the beginning of surgery, somebody was diagnosed with a tumor, first thing was to try to cut it out. And surgery was the first approach. And then as, you know, advances were made, some of those patients would end up getting adjuvant treatment with chemotherapy and or radiation therapy. The surgery alone wasn't sort of enough. That whole paradigm is really changing, and it's very exciting and leading to much better outcomes. So we now are more and more doing what's called neoadjuvant treatment. So it's the additional treatment to surgery, but coming before surgery. And this this started years ago in several areas. For example, I'm sure many people are aware with breast cancer, neoadjuvant treatment can shrink a tumor and in some women avoid a mastectomy, and instead all they need maybe is a lumpectomy, removal of the tumor itself. So it's obviously a very important and great benefit. Similar sort of principle happens with rectal cancer. These patients almost now routinely get neoadjuvant treatment to shrink the tumor before surgery, and it makes the surgery much safer and better. And really importantly, many patients are able to avoid a permanent colostomy bag, a bag on the outside that collects the stool. I mean, that's necessary in some patients, but we're able to avoid it more and more because of these treatments that happen before surgery and able to make the surgery better, safer, and better outcomes. And again, avoiding something like a colostomy. So really exciting things happening now in other areas of cancer surgery, especially in stomach and pancreas, where these same principles are being applied. And so pancreas surgery has been a big area of expertise at the MGH for really decades. And my colleagues who do pancreas surgery are really at the forefront of this work, giving patients neoadjuvant treatment and shrinking the tumors, making the surgery safer, less complications, and better outcomes in terms of recurrence and survival. And very importantly, and this is really some cutting edge work that's being done at the MGH, is essentially taking people who would otherwise not be candidates for surgery. The tumor was sort of called, kind of out of bounds. And with the neoadjuvant treatment, making them amenable to surgery, and where we can go in and remove the tumor and have the best opportunity for cure or long-term survival. So it's really an exciting area. It's being done with gastric cancer also, and also is the kind of thing that really points out the importance of being at a place like the MGH, a center with great colleagues in medical oncology, radiation oncology, because these kinds of treatments really require that collaborative, multidisciplinary effort. And that's happening on the research side also with this kind of cancer surgery. We're taking advantage of this period of the neoadjuvant treatment as patients are essentially getting treated and waiting to have their surgery with a thing called prehab. People know about, I've heard of, I'm sure, about rehab, after a hospitalization, going to a place and kind of spending a week or two or whatever it takes to get better before being able to go home. Well, there's now research being done and work being done at the MGH and other places on what's called prehab, essentially taking advantage of this period of neoadjuvant treatment with a very well-designed program of exercise and nutrition, and essentially priming patients to have the best operation and the best outcomes from the surgery. So we kind of have the motto of stronger before equals stronger after. People say that who work in the prehab area. And it's, again, another exciting thing that we do before surgery. Now, when we get to surgery itself, of course, there have been many, many advances in recent years. One of the big ones, of course, has been minimally invasive surgery with laparoscopic surgery, and now sort of the next version, which is robotic surgery. You read about and see about robotic surgery all over the place. It's not for everyone. It's not for every operation, but there are certain areas of cancer surgery where it's really proving to be very beneficial. When we as surgeons think about the abdominal cavity, there are certain places that are sort of more difficult to see and visualize and difficult to work in. And the robot with the incredible imaging and ability to use instruments that can work in 360-degree angles has benefited us in many ways in those particular areas. So in the pelvis for rectal surgery, up in the left upper quadrant for liver surgery, very important. There are certain liver tumors that are not able to be accessed well, even with open surgery or laparoscopic, but with the robot, we're able to do it. Same thing with certain pancreas surgeries in the left upper quadrant area of the abdomen. So the robotic surgery is really exciting and obviously has taken off in a big way in the last 5 to 10 years. something that's really exciting in our department, where we're truly national or really international leaders. A couple of colleagues of mine, Oz Morales and Dan Hashimoto, are looking at the idea of using artificial intelligence to both teach surgery to young trainees, but also maybe more importantly, to make surgery safer. And so sort of leveraging a lot of the information that is gained from robotic surgery and using that those data basically to develop artificial intelligence to help with surgery. And if you think, for example, when you're driving your car and if you kind of veer off a little into the other lane, you know, you get the alarm signal, the lights go off, and you quickly get back on track. And that's basically what they're developing for surgery. Seems a little crazy, but it's actually not that far away to be able to have that sort of system and to, again, help with both teaching and also making surgery safer. Finally, I just touch on some of the things that are happening with post-op care in patients, and post-op care has improved enormously. In fact, some of the most benefit that we've seen in terms of decreasing complications and ultimate survival of patients with cancer has to do with getting them through the operation and being able to get them through safely. There's been a lot of work on getting people recovered more quickly, decreasing postoperative pain, getting them through the surgery and postoperative period more quickly and back home. We have this new sort of exciting program that's essentially called a home hospital, and trying to take advantage of resources where we can get people home earlier and actually provide care like intravenous fluids, testing, other things that would, under normal circumstances, have always kept people in the hospital. And now, for some people, some patients, getting them home early, getting them in a more comfortable environment where they can be with family. And I think it really seems to improve the recovery period. We all know how important sort of the mental aspect affects the physical aspect, and certainly that's the case with post-operative surgical recovery. So this home hospital program is really a great thing that we're actually at the forefront of and doing research on as a new initiative. And finally, I'll just touch on a thing called PROMS, P-R-O-M-S, which is patient-reported outcome measures. And it seems pretty simplistic to essentially listen to patients and find out how they're doing. But for decades, we've sort of looked at surgical outcomes in more sort of objective ways, like complications, cancer recurrence, survival. Obviously, those are incredibly important, but they're not the only thing. It's about quality of life, and a lot of the things that patients have been dealing with for decades after surgery just have not been sort of communicated and understood. And so there's a big effort now to essentially get more patient input and understand what they're going through after surgery, especially with operations that can be, have quite a physical toll and change life in major ways. So again, some surgeons in our group, Liliana Bordiano for one, in the colorectal area, and others, Motaz Kadan, doing work to really understand what patients are going through and try to improve their quality of life after surgery. So with that, I think I'm going to shift it back to Jay.
Thank you, Keith, Becca, and Richard for just fantastic talk and discussion. I see we have Larry Ackman with us. Larry's been a fierce supporter of the MGH Cancer Center, and he and Ronnie, his lovely wife, have helped support the MGH New York symposium, which happens typically every year about this time. Larry, we didn't have the patience to wait for you. I apologize. But we are so glad to see you, and I'd like to open this up now for you to have some comments, if you would.
I'm glad you didn't wait for me, and I apologize for being late. I wanted to thank all of you for confirming once again my tremendous respect and faith in the excellence of the research and the medical treatment that Mass General is capable of giving. I am a cancer survivor of 16 and a half years from lung cancer. I'm eternally grateful to the hospital and to the excellent surgeons, nurses, and everybody associated with my recovery at Mass General. I also had prostate prostate surgery for cancer at Mass General, and it's my privilege to be associated with your high standards of excellence and to help out in whatever way I can to find new ways of treating cancer and to encourage the supporters of Mass General to continue and increase their support, because you're doing such important work, and it is such a privilege to be associated with your high standards of excellence.
Comments are wonderful. So now we're going to open things up for questions. If you go in the Q&A box, you can ask your question, and then we will have one of our panelists answer any issues you like to discuss.
Jay, while we're waiting for folks to populate questions, maybe I'll kind of warm us up a little bit with just maybe a question to Becca and another one to Rich. So Becca, I think everyone's well aware that the COVID pandemic had major impact on the ability of cancer centers to conduct clinical research, clinical trials in particular. Maybe you could give a little bit of a perspective about where we stand now and kind of our outlook, sort of through the winter months in terms of how we're standing up the clinical research program and trying to, if you will, insulate it from the effects of COVID.
Sure. And after my comments, Keith, I'd also love to hear about your experience with standing up COVID research itself, actually, because I think that was a huge endeavor that obviously had great impact for patients everywhere. Just in terms of the Cancer Center, briefly, at the peak of the first surge, when Massachusetts was very much in almost lockdown mode, many things at the hospital had to change because of state mandates, and clinical trials had to be dialed back a bit because of the same. And there were elective procedures were on hold. Inpatient admissions really had to be reserved for people who were coming in sick with anything, but also a lot of COVID. And so our ability to really enroll patients in clinical trials across the board, we kind of voluntarily dialed that back because we understood where the pandemic was in terms of kind of what patients needed. At the same time, I think our commitment has always been to the fact that our patients with cancer need treatment. And the Cancer Center certainly, of course, never stopped treating patients. And for clinical trials that were really what we thought were life-saving or truly disease-altering, we never stopped those either. And so I am actually very proud of the fact that we were enrolling patients in clinical trials if we thought it was really going to be a disease-altering course of treatment for them throughout the worst of the pandemic. And all of the people, from the regulatory people working on the studies, to the nurses, to the infusion nurses and the research assistants, all kind of just contributed to make that happen. I think now we're in a much better place after the worst of the peak in the early spring. We kind of bounced back, and we're pretty much at full force and growing. And our commitment now is to kind of get better drugs, get more people on studies. I think the winter may be hard, and we're looking into another surge, and we're anticipating that there may be changes as the next few months come. but we're in a very good place right now where we kind of know how to dial down and dial back up. And the commitment to really treating our patients who need their treatments, and those treatments are clinical trials for our cancer patients, that has never kind of wavered in any way. But Keith, can you comment a bit on standing up COVID research? Because I think that was a huge endeavor.
Yeah, I think, no, thanks. I'll make a brief comment that, about, what has been a really massive undertaking. I mean, hundreds of people who effectively didn't know each other have come together around COVID to conduct parallel clinical and laboratory research. And this was an enterprise that came together over the course of a couple of weeks, really, back in March, but has now been really cohesively operating as a research community now ever since then. And basically, we've taken sort of two prongs, or developed two prongs in that research program. One is to advance therapies, and leaving aside vaccines, we're involved in that research as well, but that's prevention. And we had a problem of actually just dealing with the virus and trying to very quickly strategize, as I alluded to. In cancer, we have many opportunities to pursue. Well, in COVID, even more in some ways, of proposed ideas, and we needed the strength of our scientific community and a collected group of them. We actually formed multiple groups for sort of the scientific deliberation, if you will, of these various opportunities. Which drugs we felt really most impactful, and while there were more than 120 clinical trial proposals in the first few month period of time, no institution, including Mass General, has the bandwidth to go pursuing more than 8 or 10 of those, and certainly at a time. So that's been really quite gratifying, the way in which our community came together and learned how to work together, and learned how to be really collaborative. Considering that not all ideas were going to be pursued, that meant a lot of really good ideas, very well-intentioned ideas, weren't going to move forward. And yet those same investigators would then enlist and support those studies that in fact we did participate in. We're now publishing multiple papers in the realm of understanding the biology of the disease more, understanding why it is and how it is that the immune system overreacts to the virus. And I think that's absolutely critical, as now within a year's time, we'll be launching a wave of clinical trials that follow those discoveries, those findings. So yes, we've had some early wins in terms of therapies that are helping to beat down the severity of the disease already. That's led to FDA approval, in fact, of 3 therapies, trials that we've participated heavily in from the first wave. But it's this research that we've done in terms of understanding the virus that's really going to inform the clinical trial portfolio in this second wave, which hopefully will be as far as we need to go if the vaccines can roll out quickly enough in 2021. So let me turn back to cancer real quick. One of our participants was asking a question about pancreatic cancer and what research we have going on there. So I thought, Rich, you could maybe tackle that. And there's a component of the question which I'll tackle, which has to do with collaboration with other institutions, including Johns Hopkins, where I went to medical school, and I grew up in Baltimore. But I happen to know about a research program that's actually been developed between Mass General and Johns Hopkins. So maybe I'll comment on it. But you want to maybe tackle what you see as most potentially impactful in the pancreatic cancer research area.
All right. You're asking about, the question was about pancreatic cancer, right? Yeah. That's been a real challenge. We all know, not all cancers are the same, and pancreas cancer happens to be a really tough one. And unfortunately, still, most people who are diagnosed with pancreatic cancer are kind of diagnosed too late, where it's already spread beyond where at least we can do things like typical chemo, radiation, surgery, and have a chance to cure them. So of course, one area that's people have been working on a lot is try to figure out how to diagnose patients earlier, and that's still tough nut to crack, I would say. Looking for some sort of screening test that could possibly, identify patients early on. And that's where some of the, research I think is really important, looking at genetic changes and so forth, where you might be able to get a clue early on that somebody has a early tumor before it gets too late, which is unfortunately, again, still a problem in pancreas cancer. But there are, there's a pretty big subset of patients with pancreatic cancer who now are getting great benefit from the novel approaches, better chemotherapy and targeted radiation therapy. And specifically, as I was talking about, with the surgical part, is to give that treatment up front. And it seems pretty maybe obvious because we do it in other areas, and why not pancreas cancer? But it wasn't so obvious. And pancreas cancer surgery is pretty complex. It's kind of high risk type of surgery, and frankly, it took sort of a pretty aggressive mindset to say, well, maybe we can do this. Maybe we can treat patients with chemoradiation and shrink tumors enough that we could go in and take them out where previously we couldn't take them out. And that's something that did actually happen at the MGH, the pancreas surgeons. And it's not me, it's my colleagues, Christina Ferroni and Keith Lillamo, Carlos Fernandez del Castillo, Motaz Kadan. Those are those four main ones. They really, they're excellent technical surgeons and are good enough to sort of have had that aggressive mindset and try to take patients to the operating room who previously would not have gone. And as it's now, there are data coming out and they're proving it, that it's actually benefiting people, and people are getting better outcomes, longer survival because of that aggressive approach. So still a lot of work.
Yeah, no, thanks. And I actually wanted to add to that quickly, just on this other part of this question, so that it's really, again, another element that I really like to celebrate about MGH as a research environment is the multidisciplinary clinical care and the way in which it comes over into the research domain. So this same team of surgeons and medical oncologists and laboratory scientists have come around the table together to basically take advantage of the fact that we're that's has this program whereby therapy is given before surgery, to really try to understand then before pancreatic cancers become very advanced, what are the sort of molecular threads, if you will, that we could pull to help, kind of unravel the problem. What we've learned in the past 10 years is that the types of therapies that have advanced to help other cancers, most notably cancers like lung cancer, as Becca was highlighting, melanoma, which is the cancer that I focus on in terms of an area where, again, this molecularly targeted therapy and immunotherapy have made huge advances. Pancreatic cancer has been left behind. Why is that? Well, we actually have some pretty solid leads there, but we need to understand that in patients, not in mice, not in the laboratory setting alone. We understand that in patients, and that's where a research team has come together. It's a multi-project, 4-project team of Mass General and Johns Hopkins investigators who pulled together what's a program grant, if you will, for the NIH. A proposal, but some of this pilot work has been ongoing with philanthropic support to basically really try to understand the cells that support the pancreatic cancer cells, so these normal cells, if you will, that are in the environment, that interaction, but critically within the cancer cells themselves, what our investigators and Hopkins investigators separately have identified is that there's a unique program in pancreatic cancer cells where they are able to sort of essentially live in the face of genetic alteration or genetic damage, and in a way that depends on these supporting cells. And that's an intersection that really had not been previously appreciated. And so there's this multi-project research team that's come together across the 2 institutions. It's really quite exciting. I've been asked to advise that group, so I just happened to be quite well aware of it. But I wanted to take that as a bridge over to two questions that came in regarding brain tumors, glioblastoma, and then just brain tumors more generally. As Rich was saying, pancreatic cancer is one of those cancers we have our eyes on because it really is one that has, as a cancer population, we have just not been able to make advances for those patients the way we have many other cancer types. But brain tumors are like that also, glioblastoma. In fact, I generally refer to pancreatic cancer and glioblastoma as the two that really sit in a unique position, not benefiting from these recent advances in therapies that have helped other cancer patients over the past 5 and 10 years, and where we need some unique solutions. And this is another area where if we didn't have the excellence that we have across the board, but starting with neurosurgery, and then the laboratory investigators who've been analyzing patient tumor specimens, and in collaboration with medical oncologists and neurologists who treat these patients with medical therapy and conduct clinical research, we wouldn't be in the position that we see ourselves in now of being able to subdivide glioblastoma and brain tumor populations using some of the same principles Becca described, really not considering this one disease, but letting the molecular information inform us in terms of how to break down what we thought 10, 20 years ago was one cancer into 10 different cancer populations. So it's not that we're picking a number, but just responding to the data. And then understand there might be unique opportunities for each of those. So this dividing and conquering concept, if you will, continues to spread. But the two questions there that I think touch on that theme quite nicely. Just keeping an eye on the clock here, Jay, I think I should probably turn it back to you for closing remarks. I love when we get questions like this because it reinforces the sense of shared purpose that we have with our patients. We really are trying to make advances in real time here for some of these cancers of greatest unmet need.
If I could just add a few comments about brain tumors, because that's my field. I think one major change is the redefinition of brain tumors, particularly in the pediatric population. Medulloblastoma, as an example, is not one disease. It's now broken out into 4 different diseases that therapy can be tailored, particularly in lowering the dose of cranial spinal radiation to these kids and still offering young children and adolescents a improved survival with decreased toxicity. And David Lewis, who's chief of pathology at the Mass General, just edited the 6th edition of the World Health Classification of Brain Tumors, which doesn't look anything like the 5th edition. Things aren't called glioblastoma anymore. They're all defined by sort of the molecular findings of the tumor. Unfortunately, in glioblastoma, and I'll end there, we have not made a lot of progress despite an enormous effort on our team and other major neuro-oncology centers in the United States. So it's still somewhat frustrating. In children, the picture is much brighter. The modern technologies to treat pediatric brain tumors, including targeted therapy, targeted radiation, have made significant improvements, maybe not in survival, but in quality of life. These kids can grow up with pretty much a normal life, except for some neurocognitive issues that can develop later on. But we are very optimistic, particularly in the pediatric subgroup. I don't see any other, Keith, can I ask you one more question before we wrap up? Do cancer survivors have a higher COVID death rate?
Yeah, good question. We've been studying this from the beginning, and we haven't seen that in the survivor community. We've been quite worried about patients who are actively undergoing active treatment. And so again, just reassuringly, I'd say patients who have gone through their course of treatment and are being monitored thereafter, that group does not seem to have a higher risk of getting COVID or having severe complications from COVID. But we have learned that if we are in the midst of treating patients, particularly with what we call conventional chemotherapy drugs, we've talked a little bit about that today, what that means. But drugs that temporarily suppress the immune system, that can create a problem. And we've got evidence now that those patients need to be particularly carefully watched, but also really in their households and their communities really need to exert extra precaution as they're undergoing their treatment to avoid contracting COVID in the first place. But it turns out that most of the therapies that we've talked about this morning, including the targeted therapy, molecularly targeted therapies, and the immunotherapy drugs, we've looked at those specifically, and the patients who are receiving those drugs don't seem to have any greater risk posed by COVID, which is good news. So I think we've got our spotlight set on those for whom we do need to pay the most attention.
Thank you, Keith. Again, I'd like to thank Larry and Ronnie Ackman and the organizing committee for sponsoring this event. It's always a real pleasure to come to New York, and we're not really in New York, but I'll say we're in New York right now. And hopefully next year with the vaccine, we'll be able to return to that glorious city to update what the Mass General is doing in advancing the outcomes for patients with cancer. Also like to thank Heidi Bergmeyer and Stephanie Martin and their team for helping to organize this, and to thank Keith, Becca, and Richard for taking time out of their incredibly busy schedules to present today. So I'll end it there, and thanks, and hopefully we'll be back at the Harvard Club or someplace in New York next year doing another update from the MGH. Appreciate it.